Hello, and thank you for coming to my poster today. I am Silvia Anderle. I'm a PhD student at the University of Sussex. And today I will talk to you about the first results of my PhD. So to give you a little bit of a background, APOE-4 is associated with increased risk of developing Alzheimer's disease. And it is associated with a number of deficits, which are earlier cognitive decline, blood-brain barrier dysfunction, and earlier plaque onset, and earlier neuronal deficits. Neurons, when they are activated, they need nutrients and oxygen to be active and to function properly. And these nutrients and oxygen are delivered to the neurons via the blood vessels. And when the neurons need more nutrients, then the blood vessels dilate and more blood goes through the vessels and reaches the neurons, delivering more nutrient and oxygen. And this phenomenon is called neurovascular coupling. So there is a coupling between neurons and vessels. However, previous findings from our lab suggest that APOE4 carriers might actually have impaired neurovascular coupling. And this means that not enough nutrients and oxygen are delivered to the neurons when they are active, leading to conditions that are detrimental, such as hypoxia and ultimately neuronal damage and death. But exercise, on the other hand, is known to be beneficial in maintaining vascular function and also induces angiogenesis in the capillary bed and can reverse the neurovascular deficits caused by Alzheimer's disease. And this is especially true when exercise is done at young age. Therefore, exercise might actually help to prevent and reverse the neurovascular deficits that are seen in APOE4 mice. So with this in mind, my first aim is to investigate the effect of exercise and APOE status on red blood cell velocity and capillary and pure vessel responses during neuronal activation. And then my second aim is to create a recording setup for exercise from the mouse home cage. So for my first aim, the way that I'm investigating the vascular responses in the mice is by doing a surgery over the visual cortex, which enables me to look to visualize in vivo the neurons and vessels in the mouse visual cortex using a 2-photon microscope. And I can activate the neurons by presenting the mouse with a visual stimulus. And when the neurons are activated, they become fluorescent. I can also visualize the blood vessels by injecting them with a fluorescent dye, which enables me to visualize both the capillaries, which are the smaller vessels, and the PO vessels, which are larger and more superficial. And this is what we can see. Basically, when the vessels, when the neurons are activated, the vessels respond by dilating. And we can quantify if the vessels are responsive, so if they dilate when neurons are activated, or if they are not responsive. And we can also use line scan to calculate the red blood cell velocity. And this is done by calculating the angle at which the lines are detected. So when the velocity is faster, the lines will become flatter. So if we compare the angle at which they pass through the scan at rest to the angle at which they pass through the scan during stimulation, we can then definitely calculate the change in red blood cell velocity. And this is a timeline of my recordings. I do surgery on the mice at 9 weeks, and then some of them are given a wheel and some of them aren't. And today I will present the recordings I collected at 3 months. And then I will also collect recordings at 6 months of age. And throughout my timeline, I also record exercise from the mouse home cage using a camera that is positioned at the top of the cage. And then I use this Deep Lab Cut software to label the mouse body parts and the wheel. And Deep Lab Cut trains deep neural networks to recognize the body parts. So to move on to the results, these are the average red blood cell velocity traces before and during stimulation, so when the neurons are active. And you can see that there is an increase in red blood cell velocity. So we can then quantify this increase by calculating the area under the curve, which is shown here. And we can see that the average area under the curve during neuronal activation is higher in mice that exercise compared to mice that don't exercise. And we can also look at the responsiveness of the capillaries, so their ability to dilate in response to neuronal activation. And what we are seeing is that APOE-4 mice show a significantly lower responsiveness compared to the other groups. But it appears that exercise might actually rescue this impairment, with APOE4 mice that exercise showing an increased responsiveness. And then we can also look at the responsiveness in the large vessels, which are the pill vessels. And what we are seeing here is that APOE4 mice have lower responsiveness, and this is irrespective of the exercise status. Therefore, in summary, we are seeing that APOE4 carriers have decreased responsiveness of the capillaries and PO vessels, which don't dilate as much during neuronal activation. And this situation might lead to a detrimental condition, because it is possible that neurons don't receive enough nutrient and oxygen to function properly, and leading ultimately to hypoxia or neuronal damage. But it seems that exercise might actually rescue this impairment seen in the capillaries by increasing their responsiveness, and also by increasing the red blood cell velocity responses. And in the future, what I will look at will be to assess the extent of neuronal activation during visual stimulation, and correlated this finding with the vascular values. And I will also correlate the amount of exercise that the mice do in their home cages to an index of neurovascular coupling. So these are my references. And to conclude, I would like to thank the School of Psychology at the University of Sussex, who is funding my PhD. And I would like to also thank and recognize Sussex Neuroscience. And also a special thank you to all the lab members who have provided me with practical and moral support throughout my PhD. And also a special thank to Andre Chagas, who also collaborated with me and helped me with establishing the exercise recording setup.